82 dead sheep lay scattered across the dust of a Puerto Rican farm in March of 1995, each one of them drained of blood through 3 surgical punctures in the neck. There was no struggle, no tracks of a wolf or a stray dog, and most disturbingly, not a single drop of spilled blood on the parched earth. This was the moment the world first heard the name Chupacabra, a creature that has since evolved from a local island ghost story into a global biological enigma that defies the known laws of zoology. While the public imagination often paints a picture of a reptilian alien or a mangy coyote, the reality buried in classified reports and eyewitness accounts suggests something far more calculated. We are not just looking at a predator, we are looking at a riddle of flesh and bone that appears to have been assembled rather than born. The sheer precision of the killings in those early days of the mid-90s points away from the messy reality of natural selection. Evolution produces killers that tear, bite, and consume meat. A lion or a bear is a chaotic eater. The chupacabra, however, is a biological vacuum. In the town of Canovanas, investigators found over 200 animals dead within a single year, all exhibiting the same impossible pathology. Local veterinarians were baffled to find that the internal organs of these animals had been turned to mush, yet the skin remained largely intact except for the signature triangular holes. It is as if the creature possessed a specialized proboscis capable of injecting a powerful anticoagulant and a necrotizing enzyme simultaneously, allowing it to liquefy the insides of its prey before drinking them dry. This is not the behavior of a wild animal. It is the function of a specialized tool. The timeline of these sightings coincides with a period of intense, clandestine activity in the Caribbean. For decades, the United States has maintained high-security research facilities on the island of Puerto Rico, most notably within the dense, tropical confines of El Yunque National Forest. It is a matter of public record that the Roosevelt Roads Naval Station was a hub for experimental testing, and rumors have persisted for years regarding the Advanced Research Projects Agency's interest in gene splicing during the late 20th century. If we look at the chupacabra as a runaway product of transgenic experimentation, the pieces of the puzzle begin to lock together. The original descriptions of the beast, a bipedal, 3-foot-tall entity with large glowing red eyes, quills along its spine, and the ability to leap over 6-meter fences describe a chimera. It possesses the jumping power of a kangaroo, the quills of a porcupine, and the predatory instincts of a bat. In 1996, a series of leaked documents hinted at a project involving the manipulation of the myostatin gene, which controls muscle growth, and the integration of bioluminescent markers from deep-sea organisms. This could explain why so many witnesses report the creature's eyes as having an unnatural, internal crimson glow that persists even in total darkness. If an organism was being engineered for urban warfare or covert ecological sabotage, its primary requirement would be the ability to move silently, see in the dark, and neutralize targets without leaving a messy trail of evidence. The chupacabra fits this profile with chilling accuracy. It is a biological ghost, a weapon that got loose and began to feed on the local economy. The mystery deepened when the sightings crossed the water. By the early 2000s, the chupacabra had seemingly transformed. In Texas and northern Mexico, the reptilian leaper was replaced by a hairless, blue-skinned quadruped with a jagged gait. Skeptics and mainstream biologists were quick to dismiss these as coyotes with severe sarcoptic mange. However, when local ranchers in Cuero, Texas, captured the remains of what they called the Texas Blue Dog in 2007, the DNA results were not as simple as the media claimed. While the tests showed a hybrid of coyote and Mexican wolf, the physical morphology was alien. The front legs were significantly shorter than the back legs. The skin was a leathery, elephant-like hide that appeared resistant to the sun, and the skull structure featured a set of fangs unlike any known North American canine. If these creatures are merely sick dogs, why has the phenomenon exploded in such a specific, localized time frame? Mange has existed for centuries, yet we do not have historical records of blue-skinned, vampire-like dogs terrorizing the countryside in 1800 or 1920. The sudden emergence of these mutants suggests a recent and radical shift in the genetic pool. Some researchers propose that we are witnessing the after effects of environmental contamination, a slow-motion mutation caused by the runoff of chemical testing or the disposal of experimental medical waste. But others believe the Texas variants are simply the second generation of the original Puerto Rican experiment, adapted or crossbred with local species to survive a different climate. One of the most terrifying aspects of the Chupacabra phenomenon is the psychological effect on the witnesses. Unlike sightings of Bigfoot or the Loch Ness Monster, which often carry a sense of wonder, Chupacabra encounters are marked by a deep, instinctual dread. Witnesses often report a heavy, sulfuric smell accompanying the creature, a detail that mirrors reports of high-energy laboratory leaks or the byproduct of extreme metabolic processes. In one chilling account from 1995, a woman named Madeline Tolentino described the creature's quills as changing color, shimmering between purple and green, much like the camouflage mechanism seen in cephalopods. If a terrestrial mammal has been spliced with the DNA of a cuttlefish, we are no longer talking about a freak of nature. We are talking about a masterpiece of bioengineering. Consider the statistical improbability of the blood drainage. In a world governed by thermodynamics, a predator must expend energy to hunt. To carefully drain every ounce of blood from a goat without breaking its bones or tearing its throat suggests a level of surgical intent that serves a purpose beyond hunger. Some theorists suggest the blood is being harvested for its plasma, or perhaps the creature is a biological collector for a higher intelligence. In many cases, the beast is seen in the same vicinity as unidentified aerial phenomena, leading to the interdimensional pet theory. Whether it arrived on a craft or crawled out of a petri dish in a hidden bunker, the Chupacabra represents a breach in our understanding of the natural order. It is a shadow in the brush, a reminder that the boundaries between science and nightmare are thinner than we would like to believe. As we move deeper into the dark history of this creature, we must ask, was the Chupacabra ever meant to be found, or is its existence the greatest accidental leak in the history of modern science? The transition from the lush jungles of Puerto Rico to the arid plains of the American Southwest marked a disturbing metamorphosis in the Chupacabra's physical form, but the precision of its lethality remained unchanged. If the creature was a laboratory escapee, it was one that possessed an uncanny ability to adapt its genetic expression to its environment. By the mid-2000s, the narrative shifted from extraterrestrial-looking bipedal lizards to the infamous blue dogs of the Texan brush. These were not the monsters of folklore, but something that felt uncomfortably close to a biological mistake. When a rancher in DeWitt County discovered multiple hairless, slate-gray carcasses in 2004, the scientific community scrambled to provide a mundane explanation, yet the local observers knew that a simple case of mange could not explain the heavy, overslung jaws and the lack of sweat glands in a region where the heat reaches 40 degrees Celsius. The genetic profile of these mainland specimens presents a riddle that standard biology struggles to solve. When researchers at Texas State University analyzed tissue samples from the Cuero sightings, they identified the maternal DNA as that of a coyote, but the paternal markers were a chaotic mosaic. There were traces of Mexican wolf and something else, a sequence of proteins that appeared almost synthesized. This leads to the harrowing possibility that the Chupacabra is not a single species, but a viral genetic sequence. It is a biological agent that can jump between hosts, rewriting the DNA of local predators to suit its needs. This would explain why the creature looks like a gargoyle in the Caribbean and a distorted canine in Mexico. It isn't a breeding population. It is a parasitic transformation. In 2010, a series of strange events occurred in the rural outskirts of Mexico City that reignited the theory of deliberate mutation. Farmers found dozens of chickens killed in a single night, each with a single puncture wound at the base of the skull. The spinal cords had been severed with the accuracy of a laser, yet no meat was taken. When the local authorities attempted to track the predator, they found that its scent vanished in the middle of open fields. This disappearing act mirrors the behavior of specialized military cloaking technology. If we consider the work being done in the field of synthetic biology, specifically the creation of organisms that can survive on raw hemoglobin, the Chupacabra starts to look less like a monster and more like a proof of concept for a self-sustaining, autonomous biological weapon. The history of the 20th century is littered with secret biological programs, from the horrors of Unit 731 to the clandestine nerve gas tests in the Utah desert. It is well documented that by the 1970s, the United States military was exploring the use of animals as delivery systems for pathogens. What if the Chupacabra was the next logical step, a predator designed to destabilize an enemy's food supply without firing a single shot? By targeting livestock and leaving the carcasses intact but drained, a handful of these creatures could induce mass panic and economic collapse within weeks. The fact that the first sightings occurred near the high-security military zones of Puerto Rico, such as the Roosevelt Roads Naval Station, provides a geographic anchor to this theory that is difficult to ignore. In 1997, a former technician from a private pharmaceutical firm in the United States claimed to have seen transgenic chimeras kept in pressurized glass enclosures. He described creatures that looked remarkably like the bipedal Chupacabra, noting that they were being fed a nutrient-rich blood serum. According to his testimony, these beings were part of a project titled Eugenics of the Third Domain, which sought to merge mammalian intelligence with reptilian resilience. While his claims were dismissed as the ramblings of a conspiracy theorist, the specific details he provided regarding the creature's skin, a combination of scales and fine, needle-like hair matched eyewitness reports that had not yet been made public. The mystery took a darker turn when reports of vampire attacks began to emerge from the mountains of Chile and the forests of Russia. In the year 2002, in the village of Kalama, over 600 animals were slaughtered under the cover of a thick, unnatural fog. The local police reported seeing figures that moved with unhuman speed and appeared to communicate through high-frequency shrieks that induced nausea in humans. This biological sonic weapon is another hallmark of intentional design. Nature rarely produces a predator that uses sound to incapacitate its prey, unless it is a highly specialized hunter like a bat. Yet, the Chupacabra seems to possess an arsenal of these traits gathered from across the animal kingdom. Perhaps the most chilling piece of evidence for the mutation theory lies in the behavior of the blood itself. Veterinary pathologists who examined the victims in the late 90s noted that the blood remaining in the carcasses did not clot for several days, even in extreme heat. This implies the presence of a powerful, long-acting anticoagulant that is far more sophisticated than anything found in the saliva of a leech or a vampire bat. This substance, dubbed Factor X by some independent researchers, suggests a level of biochemical engineering that points directly to a laboratory. If this creature were a natural mutation, we would see a slow progression of these traits over thousands of years. Instead, the Chupacabra arrived on the scene, fully formed, equipped with a suite of biological tools that seem to have been selected from a catalog of nightmares. As we look closer at the geographic spread, a pattern of controlled release begins to emerge. The creature appears in areas of high tension or near significant ecological sites, almost as if it is being field tested in different biomes. From the tropical rainforests to the Texan deserts and the Siberian tundra, the Chupacabra adapts, survives, and kills. It is a masterclass in survival, but it lacks the one thing every natural species has, an ecological niche. It does not eat for hunger, it harvests. It does not hide for safety, it stalks for observation. We are left to wonder if the creature is still under the control of its creators, or if the goatsucker is a prototype that has long since outgrown its leash, breeding in the shadows and rewriting its own code with every drop of blood it consumes. The final layer of the Chupacabra mystery lies not in what the creature is, but in what it is becoming. By the year 2024, the sightings have shifted from the fringes of rural farms into the digital crosshairs of high-resolution thermal imaging and global surveillance. In the deep woods of the Pacific Northwest and the remote valleys of the Andes, modern trackers have begun to record thermal signatures that defy the laws of endothermic biology. These entities appear to be able to drop their body temperature to match the surrounding environment in a matter of seconds, effectively vanishing from infrared detection. This is a level of biological thermal regulation that does not exist in the mammalian world. It suggests that the genetic experiment has entered a phase of self-evolution, where the creature is now actively editing its own genome to evade the very technology designed to find it. In 2021, a whistleblower claiming to be a former geneticist for a private defense contractor leaked a series of encrypted files detailing what he called the Chimera Protocol. According to these documents, the original 1995 outbreaks were not accidents, but intentional deployments of synthetic apex predators designed to test the resilience of local ecosystems and the reaction times of local governments. The files suggested that the creature's DNA was built on a chassis of a rhesus macaque, but heavily modified with the CRISPR-Cas9 technology of the early 21st century. Even before that technology was officially recognized by the scientific mainstream, this would mean that for 30 years, a shadow agency has been using the world's livestock as a feeding ground for a living surveillance and sabotage system that is constantly learning and evolving. The most disturbing evidence for the mutation theory comes from the discovery of what researchers call non-terrestrial protein folding, found in the saliva of a specimen captured in the Carpathian Mountains in 2018. The proteins were structured in a way that allows them to remain stable at temperatures that would normally denature biological tissue. This points toward a terrifying possibility. The Chupacabra is not just a mix of earthly animals, but a vessel for experimental synthetic life forms designed to survive in extreme conditions, perhaps even on other planets. We are looking at a creature that is more machine than animal, a biological drone that requires the high-energy nutrients found in fresh hemoglobin to keep its complex synthetic systems from collapsing. As the creature continues to appear in new corners of the globe, the official narrative remains a stubborn wall of silence or dismissive talk of mangy dogs. Yet, the mountain of evidence, the surgical punctures, the lack of blood, the impossible leaps, and the glowing red eyes points toward a truth that is far more uncomfortable. We have shared our world for decades with a ghost created in a test tube, a predator that serves as a mirror to our own scientific hubris. Whether it is a runaway experiment or a deliberate tool of shadow warfare, the Chupacabra is no longer just a legend of the night. It is a warning that once we begin to play with the code of life, we may lose the ability to control what comes out of the dark. If this descent into the shadows of biological mystery has gripped your mind, do not let the trail go cold here. There is much more to uncover in the hidden corners of our world where science and myth collide. To ensure you never miss a moment of these investigations, hit the subscribe button and turn on all notifications so you are alerted the moment a new file is opened. If you found these facts as chilling as the desert wind, leave a like on this video to support our work and share your own theories in the comments below. Have you seen something in the dark that defies explanation? Or do you believe the truth is still locked away in a laboratory vault? Join our community of seekers, and together we will keep searching for the answers they don't want us to find.